The cash crop soybeans account for the largest source currently used today for animal protein feed. It is also the second largest source we use for vegetable oil. Worldwide, the U.S. is the leader in soybean production and export. To put it in perspective, according to the USDA statistics report for 2011, the amount of soybean committed farm acreage was at 75 million acres, which made a net financial return on investment of $36.6 million. Of those 75 million, the state of Minnesota planted soybean on 7.1 million acres, which is roughly 10% of the nation's production. These acres break down to around 44 bushels per acre. Each bushel costs at around $11.94 per, which total to just a little over $525 net gain per acre. According to the Journal of Nematology, the statistical loss of soybean yield for the state of Minnesota in 2005 from root rot caused by Fusarium strains was reported to 84,902 metric tons. This disease state is called sudden death syndrome and was a loss of around $20 million and 33 bushels. No, that's not right. 31 million bushels. This was the last reported efforts from many scientists around the United States to quantify such a loss, due specifically to SDS. Considering the demand locally and globally for soybeans and their products, we must raise the question, how do we develop an environmentally sustainable solution to prevent the future loss of soybean crops? Currently under investigation in a parallel study are the possible applications of secondary metabolites from endophytes found within the soil in which soybeans are grown in. In conjunction with the baseline deficits reported by this project, together our goal is to answer the question of how to design a treatment in which timing can be an integral factor in producing positive results. To establish the most effective timing schedule, our experiment involved the measure of photosynthetic activity throughout the life cycle of the soybean plant while infected with multiple known pathogens. To record a set of measurements, we incorporated the following methods. They began with the following materials: 35 4.5-inch pots, 105 soybean seeds, and sorghum, both treated and not treated with known pathogens. These three strains of fungus, which are commonly found in soils, Fusarium oxysporum, Fusarium virgulme, and Fusarium solane, along with Rhizoactonium, which is a bacteria also commonly found within the soil in Minnesota. The environment in which the experiment was conducted was semi-controlled in an on-campus laboratory using metal halide growing lights on a 24-hour light cycle. They were also watered daily, with the exception of weekends. The seeds were planted in excess to allow for the predicted failure rate of around 10% in the successful germination of soybeans. And in the third week, we selected the most suitable plant per pot and disposed of the extras. At the 3-week point, we also obtained our first data set. Using the portable photosynthesis system, we measured multiple parameters. By mimicking the precise environmental conditions the soybean plants were growing in, we could measure the photosynthetic rate based off the input we used to mimic these conditions. The following variables were then controlled within a small chamber in which we could insert a small portion of the leaf to measure that rate. CO2 was controlled by applying an agent that removed extra CO2 from the surroundings. At the same time, we pumped into the chamber from a controlled source CO2 so that we could maintain the specified level. We also controlled flow rate, which would mimic the amount of wind around the leaf surface. This would affect a small area around that leaf that affects the rate in which it will exchange CO2 and O2 through their stoma. In conjunction with controlling the relative humidity, or H2O, via the application of a desiccant, we could maintain the environmental conditions of our semi-controlled laboratory setting. Once these variables were accounted for, we were then able to set the leaf within the chamber and since this chamber was isolated, we were able to gather the photosynthetic rate the leaf was currently undergoing. Photosynthetic activity was then recorded which is a measure of CO2 that is fixed or converted to sugar per leaf area. This method has been used many times for varying purposes, and with the exception of using the portable photosynthesis system as an instrument within the last year, many times here at Augsburg College as well. In analysis of the preliminary results, we can already identify lower photosynthetic activity across all treatments with the exception of the variant applied with the Fusarium oxysporum strain. As to why this might be, discussion brought to light the possibility that the presence of this fungal strain may induce a response in which the soybean plant increases its rate to compensate for a deficit that the fungus has to it, whether it be in its stores or some factor along the metabolic pathway of photosynthesis. Another anomaly present is the treatment in which non-pathogenic sorghum was mixed in with the soil. Considering it has produced the lowest levels of photosynthesis, we can only apply some error as the cause to this variance result. It could be from the investigators placing them in the pore slit portions of the grow system, not supplying the 5 reps with enough nutrition to germinate properly, or the least likely, in which they were all unfit individuals of soybean plants. Only repeated experiments may provide an answer to these two anomalies. At this time, the data is far from robust enough to generate any real conclusions that may or may not establish a firm timeline to identify exactly when soybean plants begin showing signs of deficiency in their photosynthetic activity. Even though some plants are showing physical symptoms of the disease states, without further investigation, we are not able to develop a timeline as of yet. Our methods are still prone to investigator error, and only repetition of the experiment will reproduce viable results to then combine with parallel treatments, such as the one discussed earlier with the endovites, to develop a means in which we can combat these pathogens in an environmentally sustainable manner. Nonetheless, these preliminary results from this first trial suggest that this path of investigation is very promising, and that at the very least, everyone involved may contribute to a future solution. My sources, floor is open for questions.